Hello, and thank you for joining us today. Our webinar will begin shortly. We'll just give everyone a moment to sign on. Please note that today's webinar will be recorded and it will be posted on the OFPA's YouTube channel, which is available to OFPA members. Give a few more moments for some additional people to log in. Okay, I think we'll get started. Welcome everyone, and thanks for joining us today for OFPA's January webinar on novel foods, covering trends, challenges, and opportunities. As some of you may know, in addition to our spring and fall events, as well as our annual social mixer, each month we host a speaker to present on a current relevant topic related to the industry. Webinars are free for OFPA members and available to non-members via donation to the OFPA. My name is Arlene Larson, and I'm on the Board of Directors with the Ontario Food Protection Association, and I will be today's moderator for the webinar. Our agenda for today includes some housekeeping reminders, our speaker introductions, a polling question for attendees, followed by today's presentation on novel foods, and of course, a question and answer session at the end. As we get started and throughout the webinar today, we will be collecting your questions that will be posted to the presenters at the end of the webinar. As I already mentioned, for members of the OFPA, a video recording will be available of this presentation and available on OFPA's YouTube channel. Please feel free to submit your questions as we go through the presentation in the GoToWebinar control panel throughout the presentation. There will be time allotted at the end for questions and answers, and for any questions that are not addressed within the hour, I will be providing you with the contact information of the speakers today. So for today's webinar, participants will gain a deeper understanding of the definition of novel foods, an understanding of the drivers for this segment, an understanding of global regulations, and some insights into the challenges and opportunities that come with this segment. And to better equip you with that knowledge today, we have presenters Dr. Jay Alappat and Kaylee Perry. Jay is the Regional Technical Director of Chemistry for North America with Merieux Nutrisciences. He received his PhD in Analytical Chemistry from the Indian Institute of Technology, Madras, and his postdoctoral experiences in food chemistry, food safety, natural products, enzyme chemistry, and molecular biology. Prior to joining Merieux Nutrisciences, Jay worked as a Senior Analytical Scientist at Natures Fynd in both quality and research development groups. to develop fungal fermented meat protein alternatives. Jay brings an experienced perspective to the safety and quality and acceptability of novel food ingredients and their impact on sustainable food production. Jay has more than 20 years of experience in analytical method development, validation, verification, and implementation. He is passionate about developing methods for the detection and quantitation of volatile and non-volatile molecules, responsible for the sensory attributes in food and food ingredients. Jay's expertise is in the extraction, isolation, purification, and identification of natural products and has years of hands-on experience in chromatographic and spectroscopic method development. Our second speaker is Kaylee Perry. Kaylee is a food safety specialist and technical manager for new business development at Merieux Nutrisciences. She oversees novel food, produce, and nut industries as a segment manager in the Pacific Northwest and East Coast. She has over 10 years of experience in food safety and quality and regulatory compliance in produce and manufacturing. Prior to her role at Merieux Nutrisciences, Kaylee was with Dole Fresh Vegetables, where she managed food safety and quality programs, including ERP applications, traceability and crisis recall management, regulatory compliance, and process efficiency programs from farm to consumer globally. Her business background provides her with a comprehensive view of the financial, logistics, and food safety challenges facing the produce and novel food industries. Kaylee received her Bachelor of Science in Food Science, Nutrition, and Chemistry from Fresno State University. She has served on various industry committees and technical working groups and has given several presentations and served as a panelist at industry gatherings. Thank you, Jay and Kaylee, for taking the time today to present on the topic of novel foods. We will get into the presentation, but first we're going to start off with a polling question. How familiar are you with novel foods? I will give everyone a couple of moments to take the time to respond. Okay. I might have moved on a bit too quickly, but the polling results are 10% say that they're very familiar with novel foods, 71% say that they are somewhat familiar, and 19% say, what are novel foods? So I'm glad for those 19% that joined us today. Jay and Kaylee will probably run through some of those burning questions that you have. So I will let the presenters take it from here.
Thank you very much, Arlene, for the introduction. So the percentage of the people who showed awareness about novel foods, this is something we generally see when we do this kind of surveys across the world. How many people are moving to novel foods, why they are moving to novel foods, how much they know about it. And normally 30 to 40% of the people have clear understanding of why they are doing what. So that's a good representation of the general population, the polling was. So before we get into the presentation, I would like to thank you all for joining us today when we discuss one of the challenging or trends of our time. And it is an exciting field. The novel foods is an exciting field. Exciting because we may be at the cusp of finding some solutions to one of the challenging questions in front of us: how to feed the 10 billion people on Earth, in our planet, in the next 2 decades. And that too sustainably, nutritionally, and safely. That's the biggest challenge, one of the challenges we are facing now. And this is a challenge also because there are a lot of things unknown in the production process. What is the safety? What is the organoleptics? What is the acceptability? What is the quality of these products? What is the technology being used? So there are a lot of questions to be answered. In the next 45 minutes, my colleague Kaylee Perry and I will walk you through what is novel foods, how it is being classified, and what are the drivers, why we are seeing what we are seeing in the novel foods. Then later, I will introduce a little bit on the various production processes and what are the things involved in the production processes in the novel food area, and what are the challenges and what are the opportunities. Later, we move into the regulations. We'll see in the regulations, we classified into 4 different regions: North America, Europe, Latin America, and Asia Pacific. And you'll also see how diverse the regulations are, the regulatory frameworks are. Then we move to novel food testing, the chemical testing, the microbiology testing, and what are the challenges facing the analyst in the novel food testing. Then we'll conclude the seminar with some of the challenges we are facing in this area and what are the opportunities in this area and what is lying ahead of us, the path forward. Now I will leave the floor to Kaylee to introduce about the novel foods. Kaylee, please.
Thank you, Jay and Arlene. Sorry, trying to go to the next slide. Okay, so as we know, especially for the 19% of you that are what are novel foods, there really isn't a clear definition of novel foods today. We have kind of broken it down into several groups from a very broad perspective globally. The very first definition of novel foods actually was in 1997, which was food resulting from a production process not used for food production within the EU before May 15th, 1997, which gives rise to significant changes in the composition or structure of a food affecting its nutritional value, metabolism, or level of undesirable substances. So it's a very broad definition, which is why I think we've kind of broken it down into these segments from a global perspective today. So 26 years later, we kind of consider novel foods to be foods from microorganisms. So fungi, algae, food from plant or plant parts from a non-traditional propagating practices, food from animal or animal parts. We're seeing a lot more food from cells, like muscle cells from animals, cell cultures, food with an intentionally modified molecular structure, which is affecting its taste, its mouthfeel, its texture, and food ingredients that are consisting of genetically modified organisms. So moving on to kind of how we've classified novel foods in North America, we've broken it down into really 4 but 5 major categories. So first we have our plant-based category, so meat and dairy alternatives from plant-based raw material. Then we have our cell-based, which is for cultured meat. So it's meat produced from skeletal muscles or muscle cells, stem cells produced in a lab. Then we have fermentation-based, so microorganisms, fungi, algae, products from a single cell organism. And then we have insect-based alternative protein, which in North America, we are seeing is more commonly used in pet food right now. And then we kind of have this hybrid, which can be a combination of multiple categories. Mainly is going to be the cell-based, introducing the plant-based ingredients, but could also include fermentation ingredients. It's kind of uncommon to see the insect ingredients and combined in the hybrid. All right, so then we have fortified foods and supplemented foods, which has been around for a long time, which now kind of falls or can fall into the novel foods category. So a fortified food is foods for those that have nutrients added to them that do not naturally occur that affects its nutritional value. So a fortified food can become or be considered a novel food when those components or ingredients that are being added to the food are considered to be a novel food. So if those ingredients are cell cultured or plant-based ingredient, the fortified food now becomes, or the supplemented food now becomes what we're considering to be a novel food. So some of these drivers for the development of the novel food industry fall under sustainability, behavioral changes, and the economy. For sustainability, the alternative protein industry really supports this goal using lower land use and less carbon emissions than in animal-based products. Some statistics out there today would say, so if you were to consume an alternative protein burger over a meat burger, that is equivalent to driving 18 less miles when you're considering carbon dioxide, greenhouse emissions, things like that. And the statistics for the ROI on beef for 1%, when you're thinking about resources used to produce traditional beef, whereas a plant-based burger uses 87% less water, 96% less land, and 99% less greenhouse emissions. For the behavioral changes, there's this term called flexitarian, which is the combination of the words flexible and vegetarian. And this is a trend that is a flexitarian diet is a semi-vegetarian plant-forward diet. So we're seeing that up to 30% of consumers are keen to transition to a diet with less or no meat in it. And then for the economy, for the plant-based foods in the United States, we are seeing that there's more than $7 billion in retail sales, according to the Good Food Institute. For the global meat alternative market, we're seeing that's going to bring in or be at over $140 billion in retail sales just within the next decade. And that would mean that for protein, 10% of the meat industry will be from a meat alternative. And then we're seeing that there has been in the last 12 or so years that there's been over $11 billion in investments just for this industry alone. So here's kind of a visual example of the economic impact when comparing insect protein versus traditional foods. So if you look in this first column, the edible share, the orange portion represents the edible portion of the animal. So as you can see, when producing traditional beef, only 40% of the entire cow is the edible share. When as you go down to the insect, 80% of the insect is actually edible. Then you move on to the second column, and this is how much it actually costs to feed the animal. As you can see, it costs a lot less food to produce an insect. And then we go into more of the economy side, how much water it costs to produce, the carbon dioxide, and the actual land usage to produce this much of the animal. So you can see that it uses a lot more water, there's a lot more carbon dioxide, and it takes a lot more land, square meters, to actually produce only 40% of the beef, and a lot less resources to produce when you can consume almost the entire insect. So when comparing the positives and negatives of the drivers for the industry, we're seeing at the initial reaction, you know, on a positive side, there are consumers with food curiosity. They want to try new things. It's exciting, but then there also is the foodnophobia side. This is different. It's weird. I'm not sure if I want to try something like this. And then we go into the food safety side. On a positive side, you know, your animals are not being pumped with hormones, things like that, that we're seeing that are concerns from the consumers today. But then there's this distrust of biotechnology. Is it safe to consume? Can I trust that this is safe? Is it dangerous? How much do we really know? From the ethical viewpoint, as I previously mentioned about the sustainability factors, those are, for obvious reasons, being seen as a positive driver. But then there's the unnaturalist. You know, people can develop a preconceived idea that because it was made in a lab or not from a traditional animal, that it might have an off flavor or taste or texture or smell, which makes it undesirable to even try, which kind of goes back to the foodnophobia. And then from an analytical perspective, this definitely provides a lot of opportunity, as Jay mentioned, and we'll get into this a little bit more further on in the presentation. But on a negative side right now is there is a lack of regulatory requirements. We don't yet understand the official methods of analysis or proficiency testing, and all that has not been done, but then entails the room for opportunity to really grow and focus on this industry. So also from the consumer perspective, they generally like to look at 3 to 4 main things when purchasing new food. You know, the first thing is always, is it safe? Are there any allergens that I need to be aware of? Is it toxic? Will it make me sick? Then we go into the acceptability portion, which is a huge topic at the end of this presentation. Does it smell rancid? How does it feel? How does it taste when I'm chewing on it? Then we go into the quality of the product. What is its nutritional value? So if intended to replace another food, it must not differ in a way that the consumption of the novel food would be nutritionally disadvantaged to the customer. If this food is intended to replace the traditional food item, it is important that the consumer is still absorbing and consuming the same nutritional value that they were with the traditional food. Then we go into labeling. So we need to be careful not to mislead the consumer when it comes to nutritional labeling, which kind of goes back onto quality and really understanding the nutrients of the alternative protein food. And then claims, you know, as the industry continues to grow, there's going to be more claims such as this product contains more X than X, like more iron than traditional beef or things like that. So we need to be really careful that we're not making false labeling claims when moving on to the labeling of these of the novel foods industry. So now that we've kind of went over the perspective, the drivers, what novel foods are, I'm going to pass it over to Jay to kind of go over the production process and where we're at with regulatory.
Thank you, Kaylee. Now we move into the production processes. So whatever the method of production, it is the insect-based, cell-based, fermentation-based, or plant-based, the first step in the process is the selection of organism or selection of the protein or selection of the insect. That is the first part. So there are a lot of regulations on that one. It has to be safe and what it can produce, what it cannot produce. So that is the first part. The second part is to enhance the protein content. We call it as a cultivation. So in the case of fermentation products, it's the fermentation to produce the product. In the case of plant-based, combining together to make enough matrix weight to create the product. In the case of insects, it is breeding and raising the insects with the feed we provide. So that is the second step. The third step is once you have enough protein, we have to formulate the final presentation, would be a meat substitute or dairy substitute or whatever it is. That is the formulation part. That is the part where we add ingredients or take off ingredients or do several processing to make sure it has the right organoleptics, sensory attributes, or the mouthfeel. Then the last part is enhance the shelf life and distribution. So we see that in the case of insects. So first, the selection, as we know, there are several insects being approved in the European Union for food. So the selection is the important one. It goes to a rigorous study to understand the safety of utilizing or putting into the market the insects. The next part is the cultivation. So the insect has to be fed properly to get enough mass to create the protein, because every insect have various percentages of protein. It could be anywhere from 30% to 76% protein, depend on the insects. Then the next part is the processing, cleaning the insects so that it is safe and it is acceptable for consumption. Then it is the distribution part. This is the general 4 steps involved in the insect-based protein manufacturing. So in every section, every stage, from selection to cultivation to the processing to distribution, there are a lot of challenges. How to make sure the products are acceptable organoleptically and the products stay longer in their the formulation. Similarly, I went a little fast. All right, now Now we go to the cell-based. Again, the first part is the selection of the cell. There are a lot of scrutiny has to be done, making sure the cell is appropriate for the production of the product. Then the cells are being provided with the nutritional inputs to grow or multiply. So once they are multiplied, they are allowed to grow in the proper scaffolding to make the right structure, the right format of the meat. And then comes to the final formulation, adding more flavors, more attributes into it, so it is acceptable organoleptically. Then it is packed into the final distribution. Now, the same case with plant-based. The first and the foremost part of plant-based protein manufacturing is to get the right kind of protein for the formulations. Once they are done, they are being processed in different ways with the incorporation of additional flavor and texture. They may go through cooking. Then the last part of that one is the processing is to extend the shelf life before it is shipped to the customers. So in all these steps, the selection of protein, we need to look for the pesticides, the mycotoxins. When these being cooked, what are the volatiles produced which can attribute or give positive or negative organoleptic attributes. So these are the challenges in the production of plant-based product. Now go to the fermentation. The key again is the selection of organism. It could be a fungi, it could be bacteria, it could be algae. So this is the first part, and they goes through the fermentation process in the media of the carbon resources. Then the protein is produced in the plenty. There may be another step, the purification of the protein or killing the organisms. Again, this offers a lot of challenges because in the production of the protein, in addition to the production of protein, there are possibilities for producing the collateral molecules. Could be mycotoxins, it could be other metabolites, or it could be nucleic acids and so on. How to control the accumulation of these molecules is important. Then it is again goes through the process of extended shelf life and cooling, packaging, and storing and distribution. So as I said, there are 4 steps involved: the selection, the cultivation, the processing, and the distribution, formulation and distribution. So now we go into the regulations. So the regulatory framework is very at the best, it is a little complicated, I would say. From the Europeans, we have the regulations have gone more clear. It is there are a lot of regulations, specific regulations for specific activities. And by coming to the North America, in Canada, regulations are clear a little bit more than United States. Then in some part of the world, the regulations are yet to get into regulatory framework. So we see the differences between various regions of the world. Let's go to why need the regulations? The first and the foremost thing to have this regulation is to ensure the high level of protection in food safety. That is the most important thing for the regulations. And at the same time, we should also understand few standardized best practices and technical recommendations have been established today. Now, it is important to have the regulations because if you are improve the potential of this industry, this novel industry, the framework is required, and it is also critical to further advance this sector. Now, currently, the frameworks lag behind consumer demand. That is the situation current. Let us get into what's going on in the European Union. As I mentioned, European Union have strict regulations for a lot of things. For example, the regulation EC 2017-2470 clearly mentioned what are the products we can put into the market. Based on EC 2015 and 2283, and the procedures for evaluating these products are in another regulation. So scientific requirement is another regulation. How the food from a third country is imported or registered in the European regulation, European Union is given in this regulation. So there are specific regulations for each and every part of novel food introduction or placing in the market in the European Union. Now, how they do that? So this is a generic slide where it shows what is considered as a novel foods. For an example, as Kaylee mentioned before, it could be a new production process, it could be a molecular structure, it could be originating from microorganism, fungi, algae, it could be plant parts, or it could be animal parts, or it could be a plant or animal tissues produced from a cell. So this is considered as a novel food. Now, who is controlling, who is regulating these products? Of course, in the European Union is the authority with the help of EFSA. EFSA gives European Union request EFSA to study the dossier, and the EFSA study the dossier using what you call Novel Foods Working Group, and they create what they call the NDA panel, and they create a scientific opinion. The scientific opinion goes to European Union, and European Union will approve based on the opinion of EFSA. Now, in European Union, in EFSA, there are two things they look for before the introduction of a novel food. One, is it safe under the proposed use? Number two, is the novel consumption is nutritionally advantageous? Remember, most of the new novel foods are going to replace our traditional food, the meat. So that is the one thing they will look for, and everything is guided by 2015-2283 in the regulation EU. So this is how it is. What is the novel food? Who is doing the regulation and how they do it? Now let us move into North America. In North America, there are 4 agencies involved. In Canada, there are 4 agencies involved: Health Canada, Food Directorate, Canadian Food Inspection Agency, Environment and Climate Change Canada. They are involved. And the regulations what controlling the introduction of novel foods in Canadian market is Food and Drugs Act, Food and Drug Regulations, then Safe Food for Canadians Act, and Safe Food Canadians Regulations. These are the regulations involved. On the other side, United States, the 2 major organizations, agencies controlling the novel food is FDA and USDA, and most of the regulations are codified in 25 CFR 9. So as I mentioned, what does it mean by a novel food in Canada? A substance including a microorganism or food that has been prepared, manufactured, preserved, or packaged by a process that has not been previously used. So this could be an organism, it could be a food, it could be a packaging process, or a food comes from a plant, animal, or microorganism. That is considered as a novel food in the Canadian context. In Canada, as I mentioned, the Canadian Health Canada and Canadian Food Inspection Agency are the 2 major agencies involved, and that will go through the studies, will go through the Food Directorate and Novel Food section, and the 3 agencies study the safety, the hazards, and the nutritional value of this product: the Bureau of Chemical Safety, the Bureau of Nutritional Sciences, and the Bureau of Microbial Hazards. And they will put together a ruling called Food Ruling Proposal. This goes back to Health Canada and approve for the placement of the novel food in the Canadian market. Now, it's all codified in Division 28, Part B of Health Canada. Now, one of the important things to note here for the Canadian regulation is most of the rulings or the laws or the clauses coming from OECD, FAO, WHO, or Codex Alimentarius Commission. So that's how the novel food framework work in Canada. Now, let's go to United States. So as I mentioned, there are 2 agencies involved in the regulation of novel foods, USDA and FDA. Now, if a product is from a fermentation or plant-based, most of the cases it goes through what we call GRAS application. The supplier, the producer, produce a dossier to get a status of generally regarded as safe, and USDA evaluate that. And if there is no question asked, if the supplier or the producer get a letter from USDA with no question asked, that become available in the United States market. In the case of cell-based product, both USDA and FDA are involved in different levels. FDA is involved in controlling the selection of cell lines, how it is being produced and how it's being manufactured, and whereas USDA look into the good manufacturing practices, how it is produced. Now, in United States context, food or food ingredients that do not have a history of use are considered as novel foods in the United States. And as I mentioned, there are a lot of regulations in the 21 CFR. The GRAS status is given by 21 CFR 170 and 570. And in the case of cell-based meat, the sterility regulation is 21 CFR 61012, and the identity requirement is 21 CFR 61018. Now let us move into the Latin America. As I mentioned before, the legal framework, the regulatory framework in Latin America is not that very clear. And there are some regulations on the GMO related food, but not much in the fermentation, plant-based or insect-based protein products. For example, Argentina, the Ministry of Agriculture, Livestock and Fisheries are involved, and the regulations are given here. For this is for genetically modified organisms leading into the food production. In the Brazil, it is the National Technical Commission on Biosafety, and these are the few of the regulations involved in the regulation of GMO-related products in the Latin America. Now let us move into Asia Pacific or Australia. In Australia, there are the two things they will look for. Number one, it is non-traditional, that is not being used in Australia and New Zealand. That is how they consider it as non-traditional, and it's also required to assess the public health and safety, which is codified in Food Standards Code of 151 in Australia. That is how it is being regulated. The novel foods are regulated in Australia. Moving to Asia Pacific, now the only country where we have the cell-based food available is Singapore Food Agency, and according to the Singapore Food Agency, food and food ingredients that do not have a history of safe use, compounds that are chemically identical to naturally occurring substances but produced through advanced technology, that is what considered as a novel food in the Singapore Food Agency. And Singapore, the Sale of Food Act in Singapore, the Wholesome Made Food Act, these are the several acts, regulatory acts in Singapore. In Australia, the regulations are by FSNAS, Food Safety Standards in New Zealand, and India it is FSSAI. In Japan, it's slightly different regulations, and it is done by Food Safety and Consumer Acceptance. So this is the situation in the Asia Pacific. Here it shows what is the difference between various countries in the world. For example, the concept of novel foods, it is very clear in EU and UK, and it is clear in Australia. In Canada, it is clear, but there is not much clarity in Japan and United States. And there are also some geographical aspects. For an example, in the United States, it is bound by the boundary marketed in the US. It is very clear. It is inside what is happening. In Canada, it is not only limited to Canada. If it is not a history of safe use outside Canada, it is being considered as a novel food. In the case of New Zealand and Australia, in Australia, it is considered the consumption of in either Australia and New Zealand is considered as a non-traditional. On the other side, in Europe, there are two rules. One is for whatever traditional, non-traditional, the European Union, and how to regulate the foods coming from the third countries. There are two different regulations in the European context. So it is taking some time to change. All right, now let us go into the novel food testing. Like any other food matrices, the first thing to be tested is the safety. Then the second part is, so we are going to discuss in the three different baskets. One is the safety test, next one is the nutrition and the quality, and the third one is the acceptability, organoleptics and sensory. These three are important in the production of novel foods. Let us see what is the normal safety testing has to be done for the novel foods. So like any other food matrix, the environmental related analytes need to be tested for novel foods too. Arsenic, cadmium, mercury, lead, dioxins, polyaromatic hydrocarbons, polybrominated diethyl ether, etc., has to be tested for the novel foods too. Now, in addition to these, there are biosynthesized toxins. Like in the case of fermentation, there is a possibility for mycotoxins and their metabolites has to be tested. There is also a possibility in the case of plant-based products and the nutrients, which is a biggest challenge facing the industry now. Now there are other residues, the nitrates, the perchlorates, pesticides. Then allergen is a challenge for all the industries, whether it is plant-based, the insect-based, cell-based. fermentation based, it is a challenge for the novel foods. Then there will be the process contaminants. As I said, the third part of the production process is the processing. The processing can lead into processing contaminants, acrylamide, furans, methyl furans, and if there are solvents being used for the extraction, there should be a solvent residues. Then there may be possibility for processing enzymes. In addition to that, there are emerging packaging issues, mineral oil hydrocarbons, PFAS, bisphenols, non-intentionally added substrates, metals, nanoparticles. Then other undesirable molecules are melamine, cyanuric acid, and biogenic amines, which are produced by the action of microbial activities, which can make the product less acceptable to the customer and reduce the shelf life of the products. So there are microbiology tests to be done, APC, Enterobacteriaceae, EZ, SRA, yeast and mold, that has to be done. The pathogen has to be done. Then other safety criteria, B series, quack, post, staphylococci, they all has to be done like what we do for any normal product. Then test for Clostridium petrifidens for as an additional safety safety criteria. Then test for Listeria species as a general indicator for the potential presence of monocytogenes. So, like I mentioned, the normal test what we do for the food matrices has to be done. In addition to that, the novel food industry has specific challenges related to pesticides, mycotoxins, etc. Now we go into the novel foods quality and nutritional analysis. So like any other food matrices, we have to do the approximate analysis, macronutrients, lipids. It's important, the analysis of lipids are important because these products are extremely processed and the lipids may be broken down, and the broken down lipid molecules can give unacceptable organoleptic attributes. So analysis of lipids is very important. Same the case with the proteins. Because these products are being extremely processed, the proteins can break down in the different locations, producing all the various peptides and amino acids. These peptides and amino acids potentially could lead into organoleptics and sensory attributes or aftertaste. So analysis of proteins is important. Then carbohydrates, micronutrients, minerals, then other molecules like choline, lutein, carnitine, microelements, then vitamins. These are the traditional analysis of all the quality nutrition analysis we have to conduct for a novel food too. Now the acceptability testing. So this is again, as Kaylee mentioned before, one of the important things to be done is to make sure these products are acceptable to the customer. If you look into the history of novel foods, one of the main challenge for the industry is how to make sure these products have the same organoleptics attributes. It has the same mouthfeel. For that process, the products are being processed in different ways. There are things a lot are being added. There are a lot of things that are removed, and that can lead into problems to the matrices. So we have to evaluate the oxidation parameters, and there will be antioxidants added into it. So we have to figure out what is the antioxidant added into it. As I mentioned before, the protein degradation can create all those organoleptics attributes, could be positive or negative. And there will be some short chain amino acids. There will be hydrophobic amino acids. There will be lipids broken down. The broken down lipids can also give into negative organoleptic attributes. In addition to that, all the shelf life analysis, because this is a processed product, we have to know what are the organic volatile molecules produced during the process of storage, and what is the kind of taste and attributes it is attributing to the matrix. Those has to be done. Then the texture measurements, the viscosity, the pH, etc., like what we do. Now, Kaylee has been our link between our testing organization and the people who want to do this kind of test. So Katie has seen a lot of people asking, well, this is the challenge. How can we address this? How can we find the molecules? How can we solve these issues? So I will leave to Kaylee to talk a little bit about what she has experienced in the market side on the various analysis.
Thank you, Jay. So expanding on everything Jay just mentioned when it came to acceptability testing, we can really connect acceptability testing to sensory. So this is where it's important to look at like the shelf life analysis and pulling data points capturing oxidation, peroxide value, TBA, free fatty acids, all the things that really affect the The protein changes, these can cause rancidity. And then falling into make sure you're capturing those texture analysis, looking at viscosity, the pH, the color, the texture. We want to make sure that the finished product has the mouthfeel that you're going for. It doesn't have the rancidity smell. And then also expanding, as Jay mentioned, with the amino acids, the organic volatiles, the hexanol, the enzymes. We know how to test for these things in current traditional proteins today. It's now understanding how do we look at them in the alternative protein segment. Where are the proteins changing? When are they changing? And are they producing the bitter off flavors or rancidity? Are they changing the texture of the product? So these are kind of the key things that we're trying to understand. And some of the biggest challenges as Dave mentioned, as I've talked with manufacturers within the industry, are, they have questions. My product has an off flavor. My product has an off taste or rancid smell. It doesn't have the right texture. So these are all of the things that we want to look at when doing that sensory analysis that I kind of just spoke about on the previous slide, and really look at the biogenetics of the molecules causing these attributes for like the frustrations that people are saying. The molecules that we know, they can be identified. We just don't know where they're coming from and at what point in the process. So that's been a big challenge. And I would say the two biggest challenges that we're seeing right now is going to be the amount of material required for testing, especially when in R&D phases, there is very limited sample availability, which makes it challenging to accomplish all of the analysis that you need, everything that Jay went over for quality, nutrition, acceptability, needing to do shelf life. It requires a lot of material. And so when they have really limited sample availability, and I've seen more on the cell culture side especially, it's very expensive to produce at this time. So having to produce it just to do the testing, has been a major challenge. So how can we reduce the amount of sample needed to complete all of these analysis that Jay spoke about? And then adding on also to the regulatory standpoint, when there has yet to be a guidance around these specific categories in this industry. We do not have official methods of analysis. We have not done proficiency testing. So, I'm seeing a lot of questions. Well, can we just do the typical nutritional testing or quality testing that you would do on traditional beef, on my cell-based beef? And that might be acceptable right now, but we don't know if those are the appropriate methods and what we should be doing. So that's why there is such an urgent need to understand and define best methods and practices for this industry. So I'm going to pass this back over to Jay to go on to some opportunities.
Okay, so as Kaylee mentioned, there are a lot of challenges in the novel food area. So there are challenges related to pesticides. For an example, due to the global climate changes, the farmers are using much more pesticides in places, and the accumulation of the pesticides is changing. That leads us to make sure we have the robust method to detect the pesticide, the right LOQ, and so on. That is with the pesticides. Similarly, with the case of heavy metals, again, the climate changes the accumulation pattern of the heavy metals. So testing the heavy metals, novel foods at the same time, in addition to that, what are happening around the world changes our testing opportunities. And similarly with the mycotoxins, the changes in the temperature, the weather, the moisture, the conditions, etc., can also lead into differences in the microbial growth and microbial contamination and mycotoxins. That can all lead to the testing challenges. Now, again, for allergens, it's a big issue facing everywhere. We do not know what are the allergens. We do not know the de novo sensitization of allergens. In addition to that, there are changes in the allergen sensitivity related to the races, various races. Something allergenic to group of population may not be analogy to a group of population in a different part of the world. So this industry being a global, that's adding additional level of a challenge to the analytical companies. So here I'm just going to talk a little bit about the organoleptics and sensory challenges. For an example, as chemists and analytical companies, we know how to detect hexanol, we know how to detect octanol, we know how to detect octinone, and we know the attributes of these molecules. Maybe hexanol is giving a grassy note, octanol giving a beany note, or octinone giving a mushroomy note. We know those kind of things. And we also know how to test the lipids. We know how the lipids are broken down. We know what are the properties of broken down lipid molecules. Similarly, we can test the peptides and proteins. We can find out, okay, are there any preference of hydrophobic peptides in a protein or hydrophobic amino acids in a peptide? We can do this information. We can detect saponins. At the same time, are we able to connect that information to organoleptics? What is the information we are able to give to the producers of these proteins so that they can control their production process to make their product more acceptable to the customers? Please also remember, all these molecules are produced by the action of a lot of enzymes. We know how to test lipoxygenase enzymes, peroxidases, lipases, and proteases. Now, are we able to connect the action of this enzyme with these molecules and how these molecules are imparting positive or negative organoleptic attributes? These are the things need to be connected to serve, to promote the product development for the novel industries. So there are opportunities for developing a unique test. Compare this, what is how it is related to the product of these molecules, how it is controlling the organoleptics and sensory attributes of the matrix. Now, there are opportunities for grouping them all together to serve this industry. So we would like to conclude the presentation today with what we have learned by analyzing or understanding the novel food industry. Now, one thing, as Katie mentioned at the beginning, there is a change. The flexitarian trend is happening, and there are a lot of investment in this area, and everybody is working hard to make sure the product is coming to the market soon. So that means it's a new industry and new lot of challenges. So there is certainly a progress from animal kingdom to meat alternative. That means alternative sources of protein. And the research, development, and marketing of production produced new and alternative pathways are on the rise. And new industry means new challenges and new opportunities. And proper labeling, whether it's ingredients, nutrition, residues, contaminants, and secondary metabolites, is critical for guiding the decision of customers like you and me. Now, the point here is there is an opportunity for the regulatory agencies, the manufacturers, analytical companies, and customer advocacy groups to come together to address the challenges of this emerging industry. So we would like to thank you all for your time and attention. Now, I believe we have time for a few questions. Arlene?
Yep, thank you, Jay. So there is still time for you to enter any questions into the chat panel. I'll start with the first one, Jay. As the industry is moving forward, where do you see the novel food sector in the next 5 years?
This is something we have observed the way it is evolved or evolving in various parts of the world. For an example, Singapore approved the cell-based meat, and it is being sold there. And the United States, in last month, we have approved, FDA has approved the cell-based meat. So cell-based meat is concerned, I believe United States will progress compared to the rest of the world other than Singapore. And plant-based food, as it is reasonably well established in the United States, with the various companies have various products, meat alternatives and dairy analogs, it is coming out well. At the same time, plant-based products are not very common in Europe. Then fermentation in the United States, again, there are a lot of companies producing fermented products that are on the shelves for fermentation from fungi, algae, bacteria, and so on in the market in the United States. Again, Europe, we don't have a lot of companies producing that. I would say it will take some time for the Europe to catch up with the fermentation products, but US will advance faster in that area. Now, regarding the insects, we have now the European Union has three, according to the regulations, there are three insects being approved for in the European market. If we have the yellow mealworm, we have the grasshopper, and as the house cricket, 3 species are identified for the novel food for insects in Europe, and we don't have anything of that kind in the United States. Because the regulations are very different for the insect area, I believe insect sector novel food will develop in Europe much faster than in any part of the world. So that is how the situation now, if I compare the plant-based, cell-based, fermentation-based, and insect-based protein development in the next few years.
Okay, thank you, Jay.
Anything else to add, Kaylee, into that?
No, Jay, I think you captured all the areas and the growth. Yeah, thank you.
Thank you.
Okay, well, we did receive some other questions in the chat box, and we do appreciate the questions. They are, however, very technical, and the answers do involve a bit of back and forth. And due to the limitations of the platform, I did send messages to those individuals, just letting them know that we can address those questions for sure offline. So with that, I don't see any other questions in the chat box at this time. I did mention that we would share the contact information of our speakers. It is displayed on the screen now. So left it up there if you want to take a screenshot. Our time is coming to an end a little bit before the hour, but on behalf of Merieux Nutrisciences, we do want to thank you for taking the time to attend today and listening to our speakers. On behalf of the OFPA, we do thank our speakers for joining us today and bringing their knowledge of novel foods to our audience this afternoon. And if there are any questions or anything that you have that you didn't have the opportunity to ask during this session, please feel free to reach out to either the speakers or to the OFPA through the info email. And for any upcoming events that you might be interested in, please take a look at the OFPA website or follow us on social media for updates. Thanks again for attending this afternoon, and we will give you about 3 minutes back to your day.
Thanks, everybody. Thank you.
Thank you.